Hello, my dear students of class 11 science. Welcome to biology class. So today we'll begin with a new chapter, anatomy of flowering plants. So in the last class we have done about the morphology of flowering plants. So today we'll discuss about the anatomy. So what is anatomy? So anatomy means the study of the internal structure of organisms. So here we are going to learn about the cell. We are going to learn about the different types of tissues. It comes under the anatomy of flowering plant. It's also called histology. Histology means the study of tissues. Okay, histology means the study of tissues. So in anatomy, what we are going to learn? We are going to learn about the structure of the tissues in case of flowering plants. We are going to learn about different types of cells. We are going to learn about different types of tissues. And generally, what is tissues? It's mainly a group of cells having the same origin and performing the same function. Okay, so what is the need of anatomy? What is the need or what is the importance of studying the internal structure of an organism, that may be a plant or of an animal? So generally I have taken few importance. First one, it helps us to solve the taxonomic problems and establish the phylogenetic relationship among the plants. Now, what does phylogenetic means? It means evolutionary relationship among the different types of species of plants. So it helps us to place the plant in a specific rank or the category or in a taxa. Not only that, it will help us to establish a proper phylogenetic or evolutionary relationship among the plants, whether the two species they are related or not. Okay, next one I have taken the importance that it helps us in studying the structural adaptations of the plants to the variable environmental conditions. So what are the particular features in a plant that will help the plant to adapt, to adjust, to grow in a different types of climatic conditions, that we can study by the help of anatomy. Okay, so these are the few significance of anatomy. Besides that, anatomy also help us in pharmacognosy, which is the branch of science which deals with the study of sources, characteristics, and the possible use of medicinal substances. So we have got different uses of anatomy, but I have taken just few examples only. Okay, so anatomy means the study of the internal structure of organism, which is also called histology, which is the study of tissues. Now generally, what does tissue means? We just go with the brief idea about tissues. Okay, though there is no need to go in details about the tissues, but just I will touch it. Okay, so that it will be easier for you. So just listen very carefully and note it down. Okay, so what are tissues actually? So I have told you that each and every organisms, maybe unicellular or multicellular, they are made up of cells, and cell is to perform the body functions. That's why you should say cell is the structural and the functional unit of living organism. So every body functions is mainly performed by the cells. Okay, so the group of cells which has got the similar functions, which have got the same origin, they usually form a tissues. So tissues are a group of cells which have got the common origin and they usually perform the same functions. So we have got two types of tissues, kindly note it down, very important. Okay, and those two tissues are meristematic tissue and permanent tissue. Note it down. Tissues has been classified into two groups, mainly on their ability to divide. Clear? Think carefully. On the ability to divide, tissues has been classified into two groups, into two types. First one is meristematic tissue, and the second one is permanent tissue. Note it down. Now, what do you mean by meristematic tissue? Okay, so these particular tissues, they are capable of dividing. They can divide, and they are present in the regions in the plants which will grow continuously. For example, the root tip, okay, the stem tips, where the plants used to grow on continuously. So on those regions, the root tips, the stem tips, we will find the meristematic tissue. And it's also of the following types. You can see over there: apical meristems, intercalary meristems, and the lateral meristems. Now, what do you mean by apical meristems and intercalary meristems. So these apical meristems and the intercalary meristems, they are placed under the primary meristems. Listen very carefully, okay? This apical meristem and intercalary meristem, they are categorized, or they are placed under the primary meristem. So we have got two types of meristem, primary meristem and the secondary meristem. Okay, so first we'll discuss about the primary meristem, then we'll go into its subtypes, apical and intercalary. So what is primary meristems? So these primary meristems, they occur in the early life of a plant, and they are responsible for the formation of a primary plant body. So those tissues occurs in the initial development of a plant. Okay, and they are responsible for the growth of a plant. They are responsible for the growth of a new parts of a plant. Clear? And there are mainly two types, apical and lateral. Now what does apical means? So apical means they are mainly present at the tip of the root, at the tip of the stem. Okay, so in the name called apical. So they are present at the tip of the root and the tip of the stem. And stem, during their formation of leaves, during the elongation of the stem, some cells are left at the stem tip. Okay, and that cells which are left at the stem tip, they will form the auxiliary bud. Okay, the next one is lateral. Then we have got intercalary meristem. Now intercalary means they are mainly found in the mature tissues, we can say. Okay, so they are found between the mature tissues, and they are mainly found in the grasses, and they can regenerate parts which are removed by the grazing herbivore, because the herbivore mainly is to feed on the grasses. So this particular intercalary meristem, they can usually regenerate those parts which has been grazed by the herbivore. Then let's go to the permanent tissues part. Okay, the name says it is permanent, it cannot divide, it has lost the ability to divide. Okay, so after the divisions of cells in both the primary and the secondary meristems, like in apical, intercalary, lateral meristems, the newly formed cells, listen very carefully, after the divisions of primary and the secondary meristematic tissues, the newly formed cells, they become structurally and they become functionally specialized. Clear? Now once they become specialized, once they have got a particular function, once they have got a particular structure, they will lose their ability to divide. So permanent tissues, they have lose their ability to divide, whereas meristematic, they can divide, but permanent tissues, they cannot divide. Okay, and mainly we have got two types of permanent tissues, simple and complex. Simple means they are made up of only one type of cells. Simple means they are made up of only one type of cells and divided into sclerenchyma, okay, divided into collenchyma, and divided into parenchyma. These are the three types of simple permanent tissues. Then let's go into the complex permanent tissues, which are made up of more than one type of cells, and there are mainly of two types, xylem and phloem. Okay, so these are the brief idea about the tissues. Now we'll go with the next topic, which is there in our syllabus, and we are going to learn about the anatomy of roots. Okay, anatomy of roots. It's in the page number 6.25. Clear? Page number 6.25. Open it, you will get the topic anatomy of roots. Okay, so let us begin with the internal structure of dicot root, that means dicotyledonous roots. Okay, so we are going to start with the internal structure of the dicotyledonous roots. So thin TS means transverse section. Remember, TS means transverse section. So we have got two type of section cutting. All right, one is called transverse section, now one is called longitudinal section. So here we are going to learn about the transverse section of a dicot root. So when we used to study a dicot root transverse section under the microscope, we will see the presence of epidermis, we'll see the presence of cortex, we'll find the endodermis, we'll see the presence of pericycle, conjunctive tissues. Then we will see the presence of pith also, this particular space over here. We will see the presence of pith also, which is well developed in case of monocot plant. Okay, compared to dicot, it is well developed in case of monocot root. Then we will see the presence of vascular bundles. That's mainly the phloem and the xylem. Here you can see the presence of metaxylem and the protoxylem. So these are the two types of xylems. Clear? So this is the structures that we used to see when we look the transverse section of dicot root under the microscope. Now we will describe each and every one. Okay, so first of all we will learn about the epidermis. Okay, epidermis part, which is also called epiblema or piliferous layer. So epidermis, it is the outermost layer of the root. Can you see this layer? This is the outermost layer of the root. So this outermost layer of the root is called epidermis. Okay, and this epidermis is mainly composed of thin-walled parenchyma cells. Clear? And the outer wall of this particular cell, they are uncutinized. And many of the epidermal cells, they will project out, they will protrude out in the form of unicellular root hair. So you can see over here that most of the root hairs usually comes from the epidermis. So most of the epidermal cells, they usually protrude out in the form of root hair. So epidermis is the outermost layer of the root and is composed of thin-walled parenchyma cells. And the outer layer of such cells are uncutinized, and many of these epidermal cells, they will protrude out in the form of unicellular root hair. Okay, so therefore this epiblema or the epidermis along with the root hair, they will help in the absorption of water from the soil. They will help in the absorption of water, mineral and salts from the soil. So this is the function of epidermis, because generally the root hair will help in the absorption of water and the minerals from the soil. So here the epidermis along with the root hair will help in the absorption of water, minerals and salt from the soil. So it is the outermost layer of the root. Then next to the epidermis we will find the second layer, which is called the cortex. So number 2 will learn about the cortex portion of the dicot root. So can you see over in the figure the cortex portion? So just below the epidermis you will see the presence of cortex. So this cortex, it forms the major part of the plant body, that is root. Okay, and is composed of several layers of round or oval thin-walled parenchyma cells. If you look at the cortex portion, it is composed of different types of round or oval parenchyma cells. So these are the parenchyma cells, which may be round in structure, which may be oval in structure, and they have got intercellular spaces. The space between the two cells, like this particular structure, can you see here? These spaces are called intercellular spaces. So in the cortex we will find the presence of intercellular spaces, and mainly the cells of the cortex, they will store the food. They will mainly store the starch grain. Okay, now next part we will talk about the endodermis. Okay, so endodermis, it is composed of a single layer of barrel shaped cells. Can you see over here this cell, the particular round cell? This particular cell is called endodermis. Clear? So don't be confused. So this outermost cell, which you can see over here, which is round in structure, this particular cell. Okay, so this particular cell, it is called endodermis, the single layer of barrel shaped cells, and we don't see any intercellular spaces in endodermis. So endodermis are made up of single layer of barrel shaped cells without any intercellular spaces. But these endodermis, they possess Casparian strip. Okay, they have got Casparian strip. Now what is Casparian strip? Now these Casparian strips, they are made up of, or they are formed by the deposition of suberin, S-U-B-E-R-I-N. Okay, so this Casparian strip, they are made by the deposition of suberin, which will prevent the wall-to-wall movement of solute and solvent molecules in the cells. Clear? Now next part we will talk about the pericycle. So just next to the endodermis, so this outer cell was endodermis, we have just seen. Okay, this outer cell was the endodermis. Now close to the endodermis we will find a single layer of thin walled parenchyma cells, okay, just lying below the endodermis. So when you look at the structure over here, you will find the presence of this structure. Can you see where these cells just below the endodermis? These cells are there. Okay, the cells are there. These cells are called pericycle cells. They just lie below the endodermis, and they also a single layer cells. Okay, so initiation of lateral root formation, vascular cambium during secondary growth takes place through these cells of pericycle. Secondary growth we will learn it in the next class. Okay, the next one is vascular bundle. So we have finished pericycle. Now let's go to vascular bundles, which has got equal number of separate xylem and phloem. Xylem, you know, will help in the conduction of water, and phloem will help in the conduction of food. Okay, now in the young root, cambium is absent. So xylem is mainly composed of protoxylem and metaxylem. So you can see over here, so xylem is composed of protoxylem, is composed of metaxylem. Now protoxylem lies towards the periphery, it lies towards the pericycle, let's say. Okay, so protoxylem is close to the pericycle, whereas metaxylem, it lies towards the center. Very carefully, protoxylem lies towards the periphery, it always lies towards the pericycle, and the metaxylem lies towards the center. So such type of arrangement of xylem is called exarch, E-X-A-R-C-H, exarch. So in exarch, the protoxylem will lie towards the periphery and the metaxylem will lie towards the center. Okay, and phloem is mainly composed of sheath tubes, companion cells, and the phloem parenchyma cells. Okay, so here you can see the presence of phloem. So these are the phloem. Okay, these are the phloem, and here you can see the xylem. So this one is metaxylem, and this one, the close to the pericycle, is protoxylem. Okay, now in between the xylem and the phloem, you can see the presence of cells, a mass of cells. You can see the presence of this type of cells. Okay, these are the cells which are present in between the xylem and the phloem. These cells are called conjunctive tissues. Okay, so mass of parenchyma cells lying between the xylem and the phloem are the conjunctive tissues. Then next one we will find the presence of pith, a very small end parenchymatous part, and generally well developed in case of monocot root compared to dicot root. Okay, so these are the transverse section of the dicot root. Clear? Now we will see the internal structure of monocot root. Okay, so here also we are going to see the transverse section of monocot root. So when we used to see the transverse section of monocot root under the microscope, what will we observe? We will see the presence of epidermis, the outer cell like that in case of dicot. Then we will see the presence of cortex. Can you see over here? We will see the presence of cortex. Then we have got this particular layer of cells which is called, try to guess, this is called endodermis. Then below the endodermis we will find what type of cells? We will find pericycle. Then we will find the vascular bundle, that is phloem and xylem, protoxylem and metaxylem. Try to recall. Then we will find the central space over here which is called the pith. So the structure seems to be different between the dicot root and the monocot root. So we have just finished the dicot root. Now we will see the monocot root. Okay, let's go. So first one, the outer part, we will find the epidermis part. So it said that it is uniseriate. Now what does uniseriate means? Write down the meaning. Uniseriate means, uni means one. Okay, so it is single layered. So it is single layered. So it is called uniseriate. Okay, it is single layered and also composed of tubular cells having no intercellular spaces and no stomata. So here also we don't find intercellular spaces in case of epidermis, in the outermost part of the root. Clear? And some of the cells of the epidermis, they used to give rise to the root hair, like in that of the dicot root. Here also if you give rise to the root hair, and function is same. The function of root hair and the epidermal cells in the monocot root is also to absorb the water and the mineral salts from the soil. Clear? Then next to the epidermis we will find the cortex part. So this part we are going to discuss this particular structure we are going to discuss. So cortex, the wider regions of the thin walled parenchyma cells with intercellular spaces. I have told you that the space between the cells are called intercellular spaces. In the epidermal part we don't find intercellular spaces, but in the cortex we will see the presence of intercellular spaces. So these are the intercellular spaces which are present in the cortex. In dicot root it was storing the food materials, so here also it will store the food. Okay, and in older roots the outer zone of the cortex they become thick walled due to the deposition of suberin, and it forms the exodermis. Now the cortex in monocot root has got three functions: absorption, storage and protection. Okay, so it will help in the conduction of water from the root hair to the inner tissues, that is called absorption. It will absorb the water from the soil by the help of root hair and will help in its conduction to the different tissues in a root. Number 2 is storage, store the food particles. And number 3, protections. It protects the older roots because it will help in the deposition of suberin. It protects the layer of cortex. Okay, so therefore it has got three functions: absorption, storage and protection. Clear? Next one we'll talk about the next layer which is close to the cortex, with the endodermis, a single layer cell which is made up of barrel shaped cells. Okay, so these particular cells you can see over here, these particular cells we are going to discuss. So these cells, okay, so this particular cell is called endodermis cells, like in that case of the dicot root. So a single layer consisting of barrel shaped cells which are thick walled without any intercellular spaces. We don't see the presence of any spaces between the cells in case of endodermis. And also the cell possesses the Casparian strips which are formed by the deposition of suberin. Okay, and this particular endodermis will regulate both inward and outward flow of the fluid. The next one, just below the endodermis we will find the single layer of parenchyma cells just close to the endodermis. Okay, but here it will lie to the inner of the endodermis. So that particular layer or cells which made up of parenchyma is called the pericycle. So thus it lies inner to the endodermis here in case of monocot root. Okay, so we will find endodermis, then next to the endodermis or just below the endodermis we will find the next layer of cells which is made up of parenchyma cells called the pericycles. It just lie inner to the endodermis. Okay, then in between the xylem and the phloem we will see the presence of what type of cells? The conjunctive tissue cells. Okay, conjunctive tissue. So here you can see the presence of metaxylem. Okay, you can see the presence of protoxylem and you can see the presence of phloem. So in between the xylem and the phloem we will see a mass of cells which is called the conjunctive tissues. Then in the center we will find the presence of pith which is well developed in case of monocot root. Okay, so it occupies the central position or the central portion of the monocot root and is composed of parenchyma cells. And this particular pith it is well developed in case of monocot root compared to that of the dicot root. Keep in mind. Okay, the next one is vascular bundles which are made of radial, polyarch and exarch, where we will see the presence of protoxylem towards the periphery and metaxylem towards the center. So here you can see over here the metaxylem, it lies towards the center. Okay, but once you look at the protoxylem, it is located towards the periphery. It is close to the pericycle, or we can see the endodermis. So pericycle are located at the periphery and endodermis at the center. So this type of arrangement is called exarch. Okay, and here these vascular bundles are organized into 8 or more alternate bundles of xylem and phloem. So you can see over here, count it: 1 2 3 4 5 6 7 8. We will find 8 metaxylem. Here also 1 2 3 4 5 6 7 8. 8 protoxylem, and let's count the phloem. You can see phloem over here. There is 1 phloem, 2 3 4 5 6 7 and 8. So we are finding the organizations of 8 or more alternative bundles of xylem and phloem in case of monocot roots. And each xylem is mainly composed of protoxylem and the metaxylem, and we have told you that protoxylem lies towards the periphery, towards the pericycles, and metaxylem lies towards the center. and such type of arrangement is called exarch and centry petals. Okay, and like that of the dicot root, here also the phloem is mainly composed of sheath tubes, companion cells, and the phloem parenchyma. Now in case of monocot root, cambium is absent and there is no secondary growth, but in case of dicot root we are finding the cambium and we are also finding the occurrence of secondary growth, but in monocot root we don't see the presence of cambium, so there is no secondary growth also. Now from your textbook, kindly go to the page number 6.28. okay, page number 6.28. go through the differences between dicot and monocot root. Is in the page number 6.28, table number 6.20, differences between dicot and monocot root. Okay, so kindly go through it. It says that in dicot root, the vascular bundles may vary from 2 to 4 or rarely to 6. That xylem and phloem will find hardly 2 to 4 or up to 6. But in case of monocot root, we are finding how many vascular bundles, how many xylem phloem? 8 or more. In dicot root, cambium was present, right? Cambium was present, and it was also involved in the secondary growth of the dicot roots. But in case of monocot root, cambium is absent. Remember this thing. Number 3, xylem vessels in dicot root are smaller in size, but in monocot root they are larger in size. Okay, and in dicot root, secondary growth occurs after the appearance of cambium. Listen very carefully. Secondary growth occurs after the occurrence of cambium in case of dicot root, but in case of monocot root, since cambium is absent, there is no secondary growth. Pith is small or absent in case of dicot root, but in case of monocot root, pith is well developed. Clear? And in case of dicot root, pericycle will give rise to the lateral roots and the cambium, but in case of monocot root, the pericycle will give rise to only the lateral roots. Pericycle will not give rise to cambium in case of monocot roots. And dicot roots, thickening of endodermis is due to the is not so prominent due to the presence of Casparian strips, but in monocot roots, the inner cells of the endodermis is considerably thickened and it gives a U-shaped appearance. So these are the few differences between the dicot root and the monocot root. Okay, so this much for today's class. In the next class we will start with the internal structure of stem, that means the anatomy of stems. Okay, so this much for today. See you in next class. Thank you.